to educator.com. We're going to continue our discussion of cell structure and function with the topic of subcellular structure. We'll start out by talking about the subcellular structure of prokaryotic cells and then go on to talk about two types of eukaryotic cells, plant cells and animal cells. Looking at prokaryotic cells, the first thing is they come in different shapes. One shape is cocci. For example, streptococcus. So cocci is plural, and this is spherical in shape. So round cells versus bacilli, which are rod-shaped, and vibrio, which are actually curved rods. They're shaped sort of like a comma. And cholera, the causative agent for the disease cholera, the severe gastrointestinal illness, is actually called Vibrio cholera. So that bacteria has this curved rod shape. So you look at a bacterial cell, you first will notice its shape, and this actually has the shape of a bacillus. It's rod-shaped. The next thing you'll notice is the cell wall. Cell walls in bacteria contain peptidoglycan. And the name tells you what it's composed of. It's composed of short peptides, so peptido, and polysaccharides. That's the glycan part of the word. And there are two general types of cell walls, gram-positive and gram-negative. Gram-positive cell walls contain a greater amount of peptidoglycan. Gram-negative cells have only a thin layer of peptidoglycan. And the names gram-positive and gram-negative come from a technique called gram staining. In gram staining, cells are stained with a purplish dye called crystal violet. The crystal violet is then rinsed off with alcohol, and the cells are counterstained with a pinkish dye like safranin. Gram-positive cells, because of their thick layer of peptidoglycan, they retain the crystal violet. They retain the purple dye, and they appear purple. So gram-positive cells on gram stain look purple under the microscope. Gram-negative cells, when you rinse the crystal violet, doesn't really rinse out of the gram-positive cells, but the gram-negative cells don't retain that purple dye as well because they just have a thin layer of peptidoglycan. So what happens is when you counterstain, the cells retain that dye and they appear pink. So gram-negative cells appear pink, gram-positive cells appear purple, and that's due to a difference in the composition of their cell walls. In addition to the cell wall, some bacteria are covered by a layer called a capsule. This is usually composed of polysaccharides. And what the capsule does is it helps the bacterial cell to evade the immune system. It's sort of a covering that allows it to slip into a host cell. Some bacterial cells have projections from the surface that are called pili. Another name for these you may hear is fimbriae. Pili or fimbriae can have a couple purposes. One is that they allow for attachment. They allow the bacteria to attach to surfaces. Also, a specialized type of pili called sex pili allow for the exchange of genetic materials. Exchange of DNA between cells. Some bacteria are motile, and one means of motility is flagella. So flagella are structures that can move the bacteria around, and Although eukaryotic cells can have flagella, they're actually a different composition and structure. The flagella in bacteria have several parts. They actually have 3 different parts. The first part is the basal apparatus. And that's embedded in the cell membrane and the cell wall. So the basal apparatus would be right around here. And that is the motor for the flagella. It contains a pump that's powered by ATP, which allows the flagella to actually move. It provides the energy for that movement. The second part is the hook. And the hook it lies between the basal apparatus and this last part, which is called the filament, and it connects the two. So the hook connects the basal apparatus and this third section called the filament. The filament is made of a protein called flagellin. And this is a part that actually moves. And there's usually one or maybe a few flagella on a motile bacteria. Okay, so we talked about the outside of the cell, the shape of the cell, looking at the internal structures of a prokaryotic cell. The genetic material is in a region called the nucleoid region. The DNA is actually circular. There can be additional DNA in circles outside this nucleoid region, and these are called plasmids. Plasmids contain only a few genes, and they're usually not essential to survival. Typical would be virulence genes, so genes that allow the bacteria to more easily or effectively infect a host. And bacteria can actually exchange plasmids. Plasmids are also very important in biotechnology. In addition, prokaryotic cells contain ribosomes. Recall that ribosomes are structures where protein synthesis takes place. Prokaryotic and eukaryotic cells both contain ribosomes, but the ribosomes in bacteria are smaller. They are 70S size, which has to do with their sedimentation. And these are found free in the cytoplasm. They're not bound. Notice that unlike eukaryotic cells, prokaryotic cells do not have a bunch of other membrane-bound organelles. So they lack endoplasmic reticulum and Golgi apparatus. They're much less complex than eukaryotic cells. They're also smaller. For eukaryotic cells, we're going to start out by talking about a typical animal cell, and then we're going to go on to look at a typical plant cell. All right, first of all, you'll notice that they have a cell membrane or a plasma membrane. And what they don't have is a cell wall. So there is no cell wall in animal cells. The plasma membrane structure is a phospholipid bilayer, and in the next section we'll talk in detail about the structure of cell membranes, the proteins embedded in cell membranes, as well as their function. Some eukaryotic cells, some animal cells, have microvilli, and these are projections. So the plasma membrane would actually, instead of just forming more of a circular shape, it's going to project out. So it's projections of the plasma membrane, and then there'd be cytoplasm in here. And the purpose of microvilli is to increase surface area. Again, form follows function. So where you'd have to think about what tissues would need to have a great surface area, and that would be tissues that function for absorption, such as the gastrointestinal tract. And actually, cells in the intestine do have a lot of microvilli, which increases their surface area and allows them to be more effective at absorbing nutrients. Okay, looking within the cell, the genetic material is contained in a nucleus. So here's the nucleus, and it has a double membrane. Within the nucleus are chromosomes. So nucleus, important points, double membrane. And it contains chromosomes. So the DNA are organized into chromosomes, and they're bound by proteins called histones. Now, items need to get into the nucleus, such as proteins, and things also need to get out of the nucleus. Since the DNA is in the nucleus, this is where DNA synthesis takes place. It's also where transcription of messenger RNA takes place. So DNA and then transcription occurs, and there's messenger RNA that needs to be transported out of the cell. And that occurs via pores. So right here are pores. And these are openings through which materials can cross into or out of the nucleus. If you look at a stained cell, you might notice that there are some dark regions within the nucleus. And these are called nucleoli, or singular here is nucleolus. The nucleolus is a region where ribosomal or rRNA synthesis takes place. It's also an area where the ribosomal subunits are assembled. So the proteins for the ribosomal subunits, there's actually 2 subunits per ribosome. The component proteins are manufactured out here in the cytoplasm, and then they are imported in through pores. So the subunits are imported into the nucleus, to the nucleolus, and then they're assembled together to make the 2 subunits, and the ribosomal RNA is added at that point. So you have the assembled subunits, they contain rRNA, and then they're exported back out to come together as one large ribosome comprised of the 2 subunits. All right, the next item we're going to talk about, the next organelle, are ribosomes. And there are actually 2 types of ribosomes, free and bound. Free ribosomes, it's just the way the name suggests, is they're out here free in the cytoplasm. And since form follows function, protein synthesis takes place on ribosomes. So proteins that are destined for the cytoplasm are made by the free ribosomes. So cytoplasmic proteins are made here. Free ribosomes are the site of synthesis of cytoplasmic proteins. Bound ribosomes, well, what are they bound to? They're actually bound to another structure called the endoplasmic reticulum. So this is the endoplasmic reticulum, all these sacs and tunnels. And these bumps you see on the surface of part of the endoplasmic reticulum are the bound ribosomes. The area of the endoplasmic reticulum that has these bound ribosomes is called the rough endoplasmic reticulum, or sometimes just RER. And the reason it's rough appearing is because of those ribosomes. Here, proteins are synthesized that are going to be headed for export from the cell. Proteins for export. And by export, it may mean they're actually leaving the cell entirely, or they may end up just embedded in the cell membrane. So proteins made by the bound ribosomes are either headed completely out of the cell, or they're going to at least end up integrated in the cell membrane. And this makes sense because the ER can then package these into vesicles, headed for another organelle we'll talk about in a second called the Golgi apparatus, and then they're ready to go for export. So again, 2 types of ribosomes. Free ribosomes make proteins that are destined for the cytoplasm. Bound ribosomes are found on the endoplasmic reticulum, and they make proteins that are destined either for the cell membrane or to be completely exported from the cell. All right, this is just the same slide continued so that we can cover the rest of the structures in here. So I just talked about the rough endoplasmic reticulum, and I focused on the ribosomes, but now talking about the endoplasmic reticulum itself. So the endoplasmic reticulum, or just the ER, there are two types. I already mentioned the rough ER. The other type is the smooth ER. Both types are composed of a series of flattened sacs and tubules, and these are often called cisterna, these tubules. Starting with the rough ER, proteins are synthesized by ribosomes on the rough ER, and then right after those proteins are synthesized, they can enter the lumen of the endoplasmic reticulum through pores. Okay, so exported proteins made here, or made in the rough ER. In addition, the endoplasmic reticulum actually makes cell membrane. So it makes cell membrane for itself. It actually makes cell membrane for other parts of the cell, other organelles, the plasma membrane, and those get packaged up in vesicles and get transported out. So a couple functions of the endoplasmic reticulum: site of protein synthesis, site of synthesis of plasma membrane components. The smooth ER has some additional functions. So lipids are made here. Those are components of plasma membranes. This also includes items such as steroids, so substances such as steroids. The smooth ER is also the site of detoxification. So the smooth ER functions for both synthesis and detoxification. Alcohol is a substance that needs to be detoxified by cells, and in particular, the liver plays a major role in detoxification. So we would expect liver cells to have a lot of smooth ER. Okay, so two types: rough ER, which functions in protein synthesis; smooth ER, where the synthesis of lipids occurs, and detoxification. So the proteins are made here, and lipids. So the substances that are made in the endoplasmic reticulum then leave through vesicles. So vesicles bud off. There's another set of sacs right here called the Golgi apparatus, or Golgi bodies. This is a series of flattened sacs which sits right next to the endoplasmic reticulum often and communicates with it through vesicles. And there are two sides or two faces to the Golgi. There's one side that's a receiving side, and this is called the cis face. So it's the receiving side. After the Golgi apparatus does its work, it sends items out from vesicles on the trans face. So sometimes people compare the Golgi apparatus to a post office because its job is to process, sort, and package materials such as proteins and lipids. So the Golgi functions to process, sort, and package items, including proteins and lipids. So what do I mean by process? Well, the Golgi does things like phosphorylate. So it adds phosphate groups, it adds sugars, so glycosylation. So some examples to give you are phosphorylation and glycosylation. And sometimes the purpose of these is to provide a signal so that the cell knows where the protein's going. So it's kind of like a label. So the protein is made in the endoplasmic reticulum. Vesicles bud off, they fuse with the Golgi apparatus on the cis face, they enter the Golgi, they're going to be modified inside. Again, a phosphate group might be added, and that particular group might let the cell know that this is a cell membrane protein so that when it buds off the transphase in a vesicle, then that vesicle will fuse with the cell membrane, and if the protein is a membrane protein, it'll stay there. If it's not a membrane protein, then it would just be secreted out of the cell, and there needs to be some sort of signal to tell the cell where a particular protein goes. Okay, so there are several other structures that we need to cover. I mentioned vesicles, and again, these are ways of the various membranes and membranous structures within the cell to communicate with each other. They're also a way for the cell to export or import materials. So those are vesicles. Large vesicles are called vacuoles. For example, this might be a vacuole. Vacuoles actually don't play as large of a role in animal cells as they do in plant cells, fungi, and protists. For example, food vacuoles provide a way for protists to eat. So a protist might engulf food. So the plasma membrane would invaginate, food particles would come in, and then the plasma membrane will pinch off and form a food vacuole with the food inside. Protists also have contractile vacuoles. And the purpose of these is to pump out water. This allows the cell to maintain its osmolarity, and osmolarity is the solute concentration of a fluid. So again, there are vacuoles in animal cells, but they play a bigger role often in other types of cells, plant cells and protists, where you'll find food vacuoles and contractile vacuoles. Another type of structure is lysosomes, which contain lysozymes. Okay. These contain enzymes. So these are just little membranous sacs that contain enzymes that break down old organelles. They break down, I'm just going to say particles, because they can break down old organelles. They also break down pathogens. Cells in the immune system, macrophages in particular, engulf and ingest foreign particles such as bacteria. Once those are ingested, a vesicle is formed, and that vesicle can actually fuse with the lysosome. And then when the two fuse together and become one, then the bacteria, the pathogen that's inside, will be digested by these enzymes. As I mentioned, old organelles are also ingested inside a lysosome, and the purpose of that is to refresh and renew the cell. So a damaged organelle can be digested, and then its component parts are actually reused by the cell. So there's a continual process of renewal going on. The environment inside a lysosome is very acidic, so the pH in there is low. And the enzymes within the lysosome function best at a low pH. This helps to protect the cell because let's say this was to burst. If the lysosome burst and it releases all these enzymes, they could just start eating up the cell. But because the cytoplasmic environment has a higher pH, the enzymes aren't going to be as active. Now, if many lysosomes burst at once and the pH of the cell overall actually decreased, then there could be a problem. But if just a single or a few lysosomes burst, they're going to find themselves in an environment where they don't work very effectively, so the cell is protected that way. There's a set of diseases known as lysosomal storage diseases, and in these, there's an accumulation of macromolecules such as proteins, polysaccharides, and lipids. And proteins, polysaccharides, and lipids are normally digested in the lysosome. But in people with lysosomal storage diseases, some of the enzymes are either absent or dysfunctional. So they end up with an accumulation of these products, and neurons in particular are susceptible to damage through the accumulation of these products, and it causes severe neurological problems. So diseases such as Tay-Sachs disease is an example of a lysosomal storage disease. Taken together, the plasma membrane, the nuclear membrane, the endoplasmic reticulum, Golgi apparatus, lysosome, all those taken together are sometimes known as the endomembrane system. And the different parts of the endomembrane communicate through vesicles. So vesicles coming from the ER to the Golgi fuse with the plasma membrane, or vesicles fuse with a lysosome or vacuole. So the endomembrane system are various structures that often work together and communicate through vesicles. And again, that includes the nuclear membrane, the plasma membrane, the endoplasmic reticulum, the Golgi apparatus, lysosomes, vesicles, vacuoles. Mitochondria are structures that are the site of cellular respiration. So cellular respiration occurs here, and organic molecules, the energy from organic molecules is used to make ATP, and ATP provides the energy for most of the cell's functions. A typical cell contains hundreds to even thousands of mitochondria. As expected, cells that use a lot of energy, like muscle cells, have a lot of mitochondria. The structure of the mitochondria is interesting. It actually has a double membrane. So when we talked about the endosymbiotic theory of cell origin in the previous lecture, I mentioned that that double membrane is most likely the result of a membrane from the original larger anaerobic bacteria that engulfed the smaller aerobic bacteria. So there's an outer membrane from one of those bacteria, and then the inner membrane that evolved from the smaller bacteria. So mitochondria have a double membrane, and the space between the two membranes is called the intermembrane space. There are folds with the inner membrane, and within those folds is a compartment called the matrix. So there's a double membrane. There's a small space between the two membranes called the intermembrane space. And then there are folds. These folds are often called crista, and the matrix, which is the compartment formed by these infoldings of the inner membrane. When we talk about cellular respiration, you will see how various enzymes and processes are carried out in certain parts of the mitochondria. Mitochondria have their own DNA. Recall that it's circular DNA like bacterial cells. They also have their own ribosomes. Mitochondrial ribosomes are 70S, so they're similar to bacterial ribosomes. And if you want to review those similarities between mitochondria and bacterial cells, go ahead and check out the lecture on comparison of prokaryotic and eukaryotic cells and the endosymbiotic theory. The final structure we're going to talk about are peroxisomes. Peroxisomes are bound by a single membrane, and they contain enzymes that detoxify substances. The ox in peroxisomes tells you how they achieve this, and they do it by oxidation. And these are found in both plants and animals. What they do is they transfer hydrogen to oxygen to form hydrogen peroxide, which is H2O2. The only problem is hydrogen peroxide in large amounts is actually toxic to the cell. So peroxisomes have additional enzymes that continue on. They remove an oxygen and release that oxygen and end up with water, which obviously is not harmful to the cell. So again, the purpose of peroxisomes is to detoxify substances. They do that through oxidation, resulting in the formation of hydrogen peroxide. In order to then detoxify the hydrogen peroxide, an oxygen is removed, which generates water. And they break down substances such as fatty acids, and they also detoxify substances such as alcohol. They also play the role in production of bile. So peroxisomes help with the production of bile, and in plants, they help convert fatty acids into sugar. These are formed by enzymes that are made in the cytosol and by vesicles that butt off the endoplasmic reticulum. So vesicles butt off the endoplasmic reticulum. The enzymes are made separately and placed into the peroxisomes, and those are formed. In contrast, lysosomes butt off the Golgi apparatus with the enzymes already contained in them. Okay, so that summarizes the many organelles and structures in the eukaryotic cell, typical animal cell. But the structures within a eukaryotic cell, in both plants and animals actually, these are held in place by cytoskeleton. Okay, so we talked about the different organelles, and within the plasma membrane is the cytoplasm. The fluid in the cytoplasm, so that the space in here is the cytoplasm, and the fluid is called the cytosol. Even though this is described as a fluid a lot of times, it actually has a texture that's more like a gel. It's not a thin, watery fluid. It's more like a gel. So within the cytoplasm are all those organelles we talked about, the nucleus and the mitochondria, the endoplasmic reticulum. The cytoskeleton holds those organelles in place in addition to supporting the cell and giving it its shape. Finally, the cytoskeleton allows for motility. Cytoskeleton is composed of various filaments, and there's three major types of filaments. The first are microtubules. The second are microfilaments. And the third are intermediate filaments. Microtubules are the largest in diameter. So talking about microtubules, they are composed of a protein called tubulin. Tubulin subunits can be added or taken off the end. So they're made of there's these individual subunits, and the cell can add onto one end, it can take off of the other end, and that allows for motility. So the cytoplasm is not just a rigid structure that doesn't move. You can think of it more like scaffolding. So if you're building a building, you have scaffolding, and when you're finished with one part of the building, you can take it down, or if you need to build a higher part, you can add to it. So the cytoskeleton works more like that. There are several important structures that are composed of microtubules. The first one is centrioles. And this picture shows centrioles. Centrioles are found in centrosomes. Their purpose is to organize microtubules. So these are one type of MTOC, or microtubule organizing center. What this is showing is division of the chromosomes. So this is a spindle apparatus right here, spindle fibers. And these spindle fibers are also composed of microtubules. So there's a centrosome out here. Within that are two centrioles organized at right angles. And here's a cross-section showing what a centriole looks like. You see that there's 9 triplets, and each of these is a microtubule. So if you took a centriole, cross-sectioned it, looked down at it, this is what you would see. Microtubules organized in this way. The spindle fibers radiate out of these microtubules and then attach to the chromosomes, and then during anaphase, the chromosomes are separated. So centrioles play a key role in meiosis and mitosis. Recall that plant cells do not have centrosomes and they do not have centrioles, but they do have microtubule organizing centers. Okay, so first structure that's made out of microtubules is the centrioles, as well as the spindle fibers. So I'm going to put spindles. Second structure is flagella. Third structure is cilia, as well as other components of the cytoskeleton. This picture here, this cross-section, shows the structure of flagella and the structure of cilia. So flagella and cilia are both organelles of motility. Some animal cells are motile. For example, sperm is motile. And the difference between flagella and cilia is that flagella tend to be longer. There's usually just one flagella or maybe a few flagella at one end of the cell. Protozoa actually use flagella for motility. Again, prokaryotes also have flagella as well, but they're made out of different molecules and they have a different structure. So eukaryotic flagella, though, are made out of microtubules. The second organelle of motility is cilia. Cells that use cilia for motility usually have many cilia. So cilia tend to be shorter and there's more of them. Cilia are very effective for moving fluids. So the airway in animals is lined with cilia. So that if you breathe in dust, other particles, the cilia on your respiratory tract keeps mucus moving out so that instead of the dust going down into your lungs, it's going to get pushed out. Some organisms, such as paramecium, actually use cilia for locomotion rather than just for pushing fluids. Okay, so microtubules composed of tubulin. Centrioles and spindle fibers are made of microtubules. Flagella and cilia are also made of microtubules. So microtubules are fundamental in allowing for cell motility. And that is the first type of filament in the cytoskeleton. The second type of filament are microfilaments. Microfilaments are composed of actin. Actin interacts with the second protein, which is a globular-shaped protein called myosin. And you might be familiar with the words actin-myosin from the study of muscle cells. So muscles use actin and myosin interaction for motility. A second type of motility that occurs is that of amoeba using pseudopods to move. So an amoeba cell actually form these almost arms or something and move around with them, and these are called pseudopods. And these pseudopods they move because of the action of microfilaments. During cell division, a cleavage furrow is formed. So the two cell, there's one cell, and then it adds on to the size of its plasma membrane. It doubles its organelles. Mitosis occurs, and then a cleavage furrow forms, and the cell divides into two. This cleavage furrow is also the result of microfilaments. Finally, cytoplasmic streaming that occurs in plant cells and fungi is a result of microfilaments. And we'll talk more about that when we talk about plant cells, but it's a circular movement within the cytoplasm. So microfilament functions. First of all, their structure. They're composed of actin, and the actin interacts with myosin to allow for contraction of muscles. The second type of movement that can occur is the formation of a pseudopod, which allows amoeba to move, as an example. Microfilaments are essential for forming the cleavage furrow for cell division. And finally, microfilaments play an important role in cytoplasmic streaming. So that was two. The third one is intermediate filaments. As expected, these are intermediate in size between microfilaments and microtubules. And by size, I mean diameter. Microtubules have the largest diameter, microfilaments have the smallest diameter, and then intermediate filaments are in between that. And there's not just a single type of subunit that these are made from. There's actually several different types of intermediate filaments, and they each are constructed from a characteristic protein. However, many of these subunits contain keratin. Keratin helps a cell maintain its shape. And provides support. And it's keratin that helps hold the intermediate filaments that help hold organelles in place. So intermediate filaments are very important in maintenance of cell shape, support, and anchoring organelles into place. Keratin is frequently found in epithelial cells. For example, our hair and our nails are formed out of keratin. Okay, so we talked about organelles found in eukaryotic animal cells. We also talked about the cytoskeleton structure. So many of these elements are the same in plant cells. So I'm going to just focus on the differences. Plant cells have a plasma membrane or cell membrane, just like animal cells. It's a phospholipid bilayer. But looking at what's different, they also have a cell wall. And bacteria have cell walls as well, but the cell wall in plants is made of a different material. The primary cell wall is made of cellulose. Cellulose is a polysaccharide, and this particular polysaccharide, cellulose, is embedded in a matrix of polysaccharides and proteins, and the purpose of the cell wall, actually multiple purposes. One is to maintain the cell shape. The other is to provide protection. The other is to maintain the turgor of the cell. Turgor is a type of rigidity that occurs as a result of water pushing on the cell wall. So this is the central vacuole, and it fills with water, especially when the cell's in a hypotonic environment, an environment where there's less solutes than inside the cell. So the cell is going to take up water, push against the cell wall, and that will make the cell rigid. Unlike animals, plant cells do not have a skeleton, so they use this turgor, or this rigidity, to maintain their shape. So if a plant doesn't get enough water, it really dries out, it starts to wilt the stem and leaves, and that's because it's not maintaining its turgor. In addition, the cell wall prevents lysing of the cell. If a cell takes up too much water and it doesn't have a cell wall, it'll burst. However, a plant cell can't take up too much water because at some point it's going to bump up against this rigid wall. So it prevents the cell from bursting. Some plant cells actually have a second cell wall called a secondary cell wall between the plasma membrane and this primary cell wall. So that is a secondary cell wall. In addition to cellulose, the secondary cell wall contains lignin. Lignin gives the plant wall even more, or the plant cell even greater strength, and lignin is found in wood. So a plant sacrifices, it's very rigid because of that, so it can't bend as much, but in return it gets protection. It maintains its shape. All right, there's a layer associated with the cell wall, sometimes called a separate component, sometimes it's considered part of the cell wall, but this layer is called the middle lamella. The middle lamella contains pectin. Pectin is very sticky. It's a polysaccharide. If you make jelly or preserves or something, you see that pectin is used as a thickener, as a thickening agent. And since it's very sticky, it helps adjacent plant cells hold together. So there will be another plant cell right next to this one, let's say right here, and they'll actually share a middle lamella. So the other cell will have its cell wall here, and then right between the two is the middle lamella. Okay, I already mentioned briefly the central vacuole. The central vacuole is quite large. It can actually take up a third or even half of the cell. The result is that since this, especially if it absorbs more water, gets bigger, the organelles are found more at the periphery of the cell. The central vacuole functions for storage. So it stores things. It takes care of waste, so waste disposal. It maintains the pH of the cell, and it has a function in maintaining turgor, as I mentioned. Again, in a hypotonic environment, which we'll discuss when we talk about plasma membranes and osmolarity, but in a hypotonic environment, in an environment where there's a dilute substance or dilute liquid outside the cell, the plant will take up more water, the central vacuole will become larger, push up against the cell wall, and the cell will be rigid. The central vacuole also takes up waste and it separates it out. It keeps it separate from the rest of the cell so that it can't cause harm. It isolates those materials. Okay, so a couple more structures on the plant cells. Chloroplasts. Chloroplasts are a type of organelle called plastids. So plastids are a set of organelles that are not found in animal cells. They're found in plant cells, but they're not found in animal cells. There are three types. The first type is leucoplasts. The second type is chromoplasts. And the third type is chloroplasts. And this picture right here is a chloroplast. And that's the type of plastid you're probably most familiar with. Starting with leucoplasts. Leucoplasts store starch. Because of that, they're found mostly in the roots of plants and in structures called tubers. So the eye of the potato, when that starts growing, that's a tuber. So leucoplasts store starch. Chromoplasts contain pigments. In particular, they contain orange and yellow pigments. So the reason a carrot appears orange is because of the pigments that are found inside the chromoplasts in the plant cell. Chloroplasts are the site of photosynthesis. So this is a chloroplast, and the fluid inside is called the stroma. So this is a fluid found inside the chloroplast. You'll notice that it has a double membrane, just like the mitochondria. Each of these membranous sacs is called a thylakoid. So each sac is a thylakoid. A stack of these together is called a granum, plural is grana. And the enzymes that carry out photosynthesis are found in the thylakoid membrane. As you'll notice, this is green, and it's because the chloroplasts contain green pigment that give parts of the plant, such as the stem and leaves, their green color. Like mitochondria, there are features of chloroplasts that are similar to bacteria and provide evidence for the endosymbiotic theory of eukaryotic origin. Like mitochondria, they have circular DNA. They have a double membrane. They divide or reproduce by binary fission, and they have their own ribosomes that are similar in structure to bacterial ribosomes. They are 70S ribosomes. All right, we're going to do a few examples now. Example 1, match each organelle with its description. So we have Golgi apparatus, mitochondria, chloroplast, lysosome, and endoplasmic reticulum. Looking down at the descriptions, the first description, the site of photosynthesis. Well, we just covered that. The site of photosynthesis is the chloroplast. So chloroplasts contain thylakoid stacked up into grana, and the thylakoid membrane contains the enzymes that carry out photosynthesis. B, contains hydrolytic enzymes which digest old organelles and pathogens. Hydrolytic enzymes are found in lysosomes. Recall that lysosomes are present only in animal cells. They are not found in plant cells. And their function is to break down old organelles so their parts can be reused, and also to destroy pathogens that have been ingested by the cells of the immune system. Okay, C, site of production of ATP from energy released from organic molecules. That is the mitochondria. The mitochondria is the site of cellular respiration. So ATP is made there, and ATP provides the energy for most functions in the cell. D, site of production of proteins destined for secretion from the cell and production of lipids and detoxification of substances. That is describing the endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes bound to it, and it's the site of production of proteins that are going to be secreted from the cell or are going to end up in the cell membrane. The smooth endoplasmic reticulum is the site of the production of lipids, and it's also the site of detoxification of certain substances. Series of flattened sacs, this is E, site of processing, sorting, and packaging of proteins made in the rough ER. It also produces polysaccharides. And this is Golgi apparatus. So the Golgi apparatus, remember, is the center where the proteins made in the endoplasmic reticulum are sorted out. They're phosphorylated or they're glycosylated, they're packaged in vesicles, and they're sent off to their destination. Example 2, what are the functions of the cell wall? Functions of the cell wall, so we talked about cell walls in plants because animals do not have cell walls. Multiple functions. One is they provide the cell with strength. They maintain the turgor of the cell. They prevent lysis, so they prevent the cell from bursting when it's in an environment where it might absorb a lot of water. And if it overabsorbs water, it'll lyse. If it absorbs, instead of lysing though, if the plant absorbs water in its central vacuole, that is just going to push against the cell wall and maintains the cell's shape and rigidity and maintain the shape of the plant through turgor. So strength, turgor prevents lysis, and it also provides protection. What are the primary and secondary walls of cell walls of plants composed of? The primary cell wall is mainly made out of cellulose, which is a polysaccharide, and it's embedded in a matrix of various polysaccharides and proteins. Secondary cell walls also contain cellulose, and they contain a second polymer called lignin, which give them additional strength. What are the cell walls of fungi primarily composed of? Fungi have cell walls as well, but these are composed of chitin. Example 3, name 3 structures composed of microtubules and list their functions. So we talked about multiple structures that are made out of microtubules. The first one was centrioles. Centrioles help to organize microtubules within the cell. They are found in the centrosomes, and the spindle fibers made out of microtubules radiate out from the centrioles and then separate the chromosomes during mitosis and meiosis. Remember that microtubules are very important for motility of the cell. So the second structure that is composed of microtubules is flagella. Flagella allow cells to have motility. They move back and forth by an undulating motion. Again, an example would be the motility of sperm cells. Third, cilia. These also allow for motility. They are shorter than flagella, and there's often many per cell. They're especially effective in moving fluid. So they're found in areas like the respiratory tract. Also, you should be aware that there's another type of cilia called primary cilia. These are found in almost every cell in an animal, in vertebrates. However, there's only one per cell. The important thing is that these are non-modal. So they're different than regular cilia in that they are non-modal, and they have different functions. The primary cilia actually function for sensory reception and signaling pathways. They also have a different microtubule arrangement than that of cilia and flagella. So centrioles and spindle fibers, flagella, cilia, and also primary cilia, which have different functions and structure, but are still composed of microtubules. What are microfilaments composed of? They're composed of actin. And remember that actin interacts with myosin to allow for cell motility. And now we're asked to give two examples in which microfilaments are involved in cell motility. One of these is muscle contraction. So the interaction with actin and myosin allows for muscle contraction. Second would be cytoplasmic streaming. Cytoplasmic streaming is found in plant cells and fungal cells, and let's say in a plant cell, what will happen is the cytoplasm will actually flow around in a circular pattern. And you can think of it as analogous to stirring up something when you're cooking. And the purpose is actually to move things around. This allows for nutrients to get where they're needed, for exchange of materials between the organelles, instead of the cytoplasm just sitting there. By sort of stirring it up and moving it around, it allows for exchange of materials. So cytoplasmic streaming in plants and fungi are a result of microfilaments. A couple more. I mentioned that amoebas move using pseudopods. And these are the result of microfilaments. That's what allows the pseudopods to move. Finally, cell division, the cleavage furrow. When one cell is preparing to divide into two, it forms this furrow and then separates off into two cells. Example 4. Why do some antibiotics that target bacterial cells also affect mitochondria and chloroplasts? So it is true that there are antibiotics that are meant to just target bacterial cells, but they have also been shown in the lab to affect mitochondria and chloroplasts. And the reason for that is because chloroplasts and mitochondria have structural similarities to bacterial cells. So similarities between bacteria and mitochondria and chloroplasts. That would be a reason. For example, the ribosomes. If an antibiotic is targeting bacterial ribosomes, these are 70S ribosomes. So I'd expect the antibiotic to affect the bacteria and the mitochondria and chloroplasts, but it's going to leave the other ribosomes in the eukaryotic cell alone because those have a different structure. They're 80S. Other similarities, remember, are the DNA. It is circular. The DNA of mitochondria, bacteria, and chloroplasts are all circular. Size. Mitochondria and chloroplasts are smaller. Eukaryotic cells are larger. So these similarities could cause an antibiotic to affect all three cell types. How is this relevant to the endosymbiosis theory of eukaryotic origin? Well, the endosymbiotic theory states that organelles from the eukaryotic cell are a result of a relationship and the engulfment of smaller aerobic bacteria by larger anaerobic bacteria. So there was a larger prokaryotic anaerobic cell. It engulfed a smaller prokaryotic aerobic cell. And eventually, over time, evolution, this became a eukaryotic cell, and this smaller bacterial cell became mitochondria, chloroplasts, organelles within the cell. And the fact that mitochondria and chloroplasts are similar to bacterial cells provides support for this endosymbiosis theory of eukaryotic origin. Thanks for visiting educator.com, and that concludes this lecture on subcellular structure.